Okay, well, everyone, thank you for coming. So everybody online, thank you for tuning in. So today, the defense of Zachary Bartsch. He's from Maple Grove, so he's a city kid, but he grew up with strong roots in North Dakota. He spent a lot of time at his grandparents' farm in Buxton, North Dakota, just north of Fargo. And he claims to be originally, where his ancestors originally settled from Norway. Okay. True. And then, so then he helped take over the farm with his dad. He still has a strong connection to agriculture, which is an attractive trait of Zach, of course. And his undergrad degree was in NRM here at NDSU, and he started this grad program about 19. And so when COVID hit, it was sort of like when he was trying to finish some of his research and things like that. So he had a greenhouse project started, and we had to sort of cut it short a little bit. But to his fortitude, he started a career with Wenck, which is now Stantec, as a soil scientist. One of his accomplishments that he's pretty proud of is he led a 20,000 acre soil survey project in southwest Wyoming. And he's done quite a bit of work in western North Dakota, wetland mitigation projects, and will be working towards his professional soil classifier degree in North Dakota, but also his septic system siting and wetland delineation in Minnesota. So Zach, the floor is yours.
Thank you. And thank you everyone for coming today online and in person. Taking a lot of work to get here, and I'm excited to share it with you guys.
That one light, there's another light that's up. Yep. And the other one too.
Before I get started, I want to share a quote that I read recently, and the quote is, I can get this to click. 10,000 hours is the number of greatness, and it's by Malcolm Gladwell, meaning it takes about 10,000 hours for someone to become an expert in their field or to perfect their skill they're trying to work on. Like I said, it took me a long time to get here, and I'm not saying that I'm an expert, but when I read this quote, I couldn't help but think about soil, and specifically how long or how time works with soil. 10,000 hours isn't very long for a soil. For perspective, in North Dakota, the oldest or the youngest soils here are about 10,000 years old. And it's a product of, over this time, many cycles and years of microbial activity that it takes to produce this media that we depend on for growing our crops and to provide a living to our landowners and farmers. And humans have kind of a dichotomous relationship in that our actions, no matter that it took millions and millions of hours for soil to be formed, our actions and our disturbances can instantly erase a lot of that work and just start that clock over. So with that in mind, that's why I'm here today. I'll be talking about soil biological recovery in soils disturbed from oil and gas activity in western North Dakota. My thesis is broken into 3 chapters. The first chapter is a literature review going over the novel use of biostimulants in reclamation. The second chapter is a field study on thermally desorbed subsoil and tracking microbial recovery and biological property recovery in that soil. And the third chapter is a greenhouse study using biostimulants in kind of a novel type of study with biostimulants using blended soils. I will be starting with chapter 2 and then moving on to 1 and 3. So picking up where I left off with disturbances, specifically in North Dakota, since 2005, there's been an increase in oil and gas production as a result of increased technology for extraction, but also increased demand. And it peaked around 2019 and dropped off through the pandemic, but in the past couple of months, there's been an upward trend. In order to support that increase in production, there has to be an equal increase in the infrastructure to extract it and to transport it. Examples up here are well pads, the actual well that pumps out the oil or natural gas, and holding tanks and pipelines. Specifically, since that 2005 time window, there's been over 13,000 additional wells installed, and also currently, collectively in North Dakota, there's 29,000 miles of pipeline. These infrastructure require soil disturbances in order for them to be installed. And at some point in different time windows, there's going to be a time when you need to reclaim and replace and restore the land to its natural or pre-disturbance productivity. But it's not that easy. There's common times a lot of problems that result from reclamation, even with the best intentions or efforts. And again, just to visualize what this might look like, there's pipeline right-of-ways that have problems with salinity that inhibits vegetation reestablishment on the right-of-way. Well pads can be severely compacted and reduce microbial activity and soil function. And you might have contaminants that leak out from pipelines or from your storage tanks. And it can be further complicated when you consider the amount of farmland that there is in North Dakota. Here's a figure with the formations where oil and gas is found in North Dakota, and the dark shadowy parts throughout the state are prime farmland and farmland of statewide importance. The pipelines, major pipelines that are in North Dakota, often crisscross right across farmland and just can affect the productivity of those soils. This image is inspired by Nick Berkheimer. I use squares instead of hexagons, but the pink areas that you see have densities of oil and gas wells of up to about 2 per square mile. And it occurs in and along these areas where there is fertile farmland. And this last one is brine spills. Each ring represents 5 reported spills in that 20 square mile area. And I'm not here just to say, look at these cool facts, look at this cool map of soil. This is real land that's owned by real people. And when there are disturbances and impacts to their land, it affects the productivity for many decades potentially, and it impacts their lives. And some of the landowners feel responsible for taking care of the land that their ancestors settled on. And I'm a farmer in North Dakota, and I can relate to that. And I'm not only here as a researcher, but to hopefully improve the reclamation of any landowner's land that has been impacted. Sometimes those landowners are named Patty and Steve Jensen, and they happen to live in Tioga, North Dakota. In 2013, those landowners found an oil spill on their land harvesting wheat, and it resulted, and I'm sure it changed the whole trajectory of their lives. This oil spill occurred from a pipeline and a hole the size of a dime. It released over 21,000 barrels of oil, which is about the equivalent of 30 rail cars. And it was immediately thought to be the size of about 15 acres for a total remediation footprint. But over the course of the remediation project, it ended up being about 77 acres. And this soil was contaminated with petroleum hydrocarbons. And so to treat this contamination, the researchers on the site, Tom Sutter, used a process called thermal desorption, which involves placing the contaminated soil into an oven-like burner. It heats it up to about 350 degrees Celsius for 10 to 15 minutes, and it neutralizes the contaminants to a level that is relatively safe or non-existent, and the out product is clean soil ready for reuse. But using this remediation strategy, there was a lot of unknowns with how it affected soil properties. So over a series of studies with Tom's team, they learned that the biggest impact was soil organic carbon decreased over 30%. And thermal desorption also caused some hydraulic properties to change. Specifically, the saturated hydraulic conductivity increased with the magnitude of around 400%. Additionally, some of the textural properties changed, so the soil that was originally a loam exhibited properties more similar to a sammy loam. In further studies, they found that the ability of the soil to bind to phosphorus was increased as well, but with adequate fertilizer management, this wasn't an agronomic concern. With all this progress of connecting thermal desorption to soil properties, there were still limited opportunities to connect it to biological functioning, and more specifically, biological recovery in this disturbed soil. We care about soil biological properties because it's an important pillar in the overall reaching of soil health or soil functioning. Processes such as nutrient cycling carried out by microbial organisms in the soil was impacted, and the recovery is important for rebuilding organic matter, the organic carbon in the soil, and they can form mutualistic relationships with the plants and microbes, specifically arbuscular mycorrhizal fungi, which we will be talking about here today. And so with that in mind, if you can't restore the soil biology, the concern is that you won't have a fully functioning soil. And because the goal of reclamation is to return the land, the disturbed land, back into one that is productive, incorporating the recovery of biological properties is important. So we conducted a 4th year of this study, the 4th year of crop production in 2019, and our objectives were to investigate how the soil treatments affect grain sorghum growth, quantify how the remediation may be affecting AMF or the fungi, the mutualistic fungi, may be influencing root colonization, and determine how soil microbial abundance and other properties are affected by these disturbances. We used a randomized complete block design with 5 soil treatments and 3 replications. The soil treatments included the uncontaminated topsoil that was originally taken from the remediation site, a thermally desorbed subsoil, which was excavated from anywhere below the topsoil or the top 15 centimeters, the SP, which is contaminated subsoil that was non-thermally desorbed, and these subsoil treatments were also combined 1-to-1 with the topsoil to make TDA and SPA. There was a paired treatment effect as well with compost and manure, where one soil mixture in a replication would receive 40 megagrams of compost and manure, and then one would not. This is what the research site looked like. I just picked out some of the different soil treatments to see the differences in colors, which are characteristic of its soil properties, mostly organic matter, and the thermally desorbed soil kind of resulted in this reddish color, and it's also because of it being a parent material and containing different properties. The black at the bottom of the picture is the compost manure. Our crop rotation included hard red spring wheat in 2016. In 2017, there was field peas, hard red spring wheat again, and then grain sorghum in 2019. In order to grow the grain sorghum, we had to fertilize, picture by Kevin Horsager, and we had normalized fertilizer rates applied to all the plots to reach 101 kilograms of nitrogen and 90 kilograms per hectare of phosphorus. We incorporated it the next day, seeding in the sorghum at 9 kilograms of seed per hectare. There was a post-emergence herbicide treatment to treat the kochia out there. And at the 3rd vegetative leaf stage, we collected root samples and then rhizospheric soil in a quantity of about 1,000 cubic centimeters. The live roots were cut from the plant and preserved in formalin acetic acid, and from the soil samples, 10 grams were taken and freeze-dried. At harvest, the mature sorghum was hand harvested from 1 square meter quadrats, and the grain heads were clipped from another square meter quadrat, bagged. The above ground biomass was put into bags and weighed in the field, transported back to Fargo, and then dried in a forced air oven and weighed again, while the grain heads were dried to less than 12% moisture. They're threshed, cleaned, and then weighed for final grain yield. And just some information about our growing season. It was abnormally wet and abnormally cold for Western North Dakota, where it's characteristically hot and dry. And that comes into our observed results, but I just wanted to put this information up to show that it was 2.5 degrees cooler and almost 400 millimeters of precipitation above the 30-year average. For the root colonization, roots were cut, the preserved roots were cut into 1 centimeter segments, they were cleared, and then they were stained. And the staining allowed me in lab to see their structure and their biomass in the plant roots in the microscope. And so 10 segments were ultimately mounted onto slides, and it's hard to see, but there is a little diagram here. Maybe it's just from my perspective, but 10 segments were placed on the slides, and 5 passes were taken over each root segment. And at where you intersected the root in your field of view, you marked the absence or the presence of AMF biomass, and ultimately added up the total number of presence points and divided by the total number of observations, which was 50. And there are some examples of what you might see under the microscope with spore and their hyphae, which are their root-like structures that are useful for acquiring phosphorus in the soil for plants. For the microbial abundance, the freeze-dried soil samples were shipped and sent to Microbial ID, where they used a lipid extraction and gas chromatography and mass spectrometry method, which ultimately provided us numbers with their phospholipid markers per microbial group and a total abundance of their biomass observed in the whole soil sample or per gram of soil. The statistical analyses conducted on these data was a 2-factor analysis of variance, or ANOVA, at the alpha of 0.05. We also did a principal components analysis in JUMP, and then a linear regression on some more biological metrics. To get right into the results, the grain yield off the bat was not significantly different between any of our interaction terms or soil or compost and manure. And this is most likely going back due to the growing season, which was abnormally wet and abnormally cold, and it delayed the maturation of the sorghum. The grain heads were inconsistently along their maturing process, and so when it came to cleaning them and drying them, it resulted in the large standard of deviation that we see with the yield. But looking at biomass, we can see right away that the 40 megagrams per hectare of compost and manure significantly improved the biomass production in our study. And any of the blended treatments were significantly improved and similar to the topsoil control in producing biomass. When we look at the subsoil treatments, we saw that the TDU soil, the thermally desorbed contaminated subsoil, produced almost double that of the SP, which after 4 years, the crude oil contamination in both treatments should be below levels that have been observed to affect sorghum growth. So when you see differences in biomass production, you might naturally look at the chemical or nutrient availability in the soil. But looking at these, we saw that between the 2 subsoil treatments, there were no differences between soil organic carbon or organic matter and the amount of phosphorus in the soil. So we kept looking at our data, and we were looking at root colonization data, knowing that the level of colonization or how plant and the symbiotic fungi interact plays a part or is dependent on phosphorus availability in the soil. We see that the blended treatments again were similar to the topsoil, indicating relative reclamation success relative to the topsoil. But again, at the root colonization percentages, we saw that there's a difference between the SP and the TDU. And oops, hang with me here. So thinking about our growing season and thinking about how thermal desorption may have affected the hydraulic properties in the soil that was treated, we remembered that there was an increase in saturated hydraulic conductivity and a shift in textural properties that acted more like a sandy loam than a loam. And thinking about the increased precipitation, conceptualizing these, we concluded that with the increased rainfall, more phosphorus that was applied as fertilization was able to transfer fertilizer through the root zone at a higher rate compared to the SP. The SP did not have an increase in saturated hydraulic conductivity, and so increased precipitation or events of intense precipitation may have resulted in the water to pond at the surface or to potentially run off and transport phosphorus away from the site. And this was also reinforced with looking at the biomass production, because with increased phosphorus potentially moving through the root zone from the increased rainfall or the ability of rainfall to move through the soil surface, the increased phosphorus also would have resulted in increased biomass production, which we did see in the TDU treatment. Going on to the microbial abundance, it was evident that the topsoil concentration was driving their relative abundances, in that the largest, the microbial abundance was the most abundant in topsoil treatments. But we did see some hopeful findings that in the SPA and in the TDA, they were producing similar microbial biomass levels to the A, which indicates that this 1-to-1 mixing ratio could be a potential tool in improving reclamation success when it comes to biological properties. In the subsoil treatments, we can see that they had similar abundances between overall total microbial abundance, but also in the soil bacteria and in the gram-to-gram ratio. And overall, the manure treatment was only significant in the actinomycetes, and additionally, all soil treatments except for the A topsoil had reduced abundance of actinomycetes, suggesting that their abundance or their ability to recover was impeded by these disturbances at this site. In our PCA analysis, we were using this to analyze how different microbial or biological properties were interacting with each other. And on the x-axis, you can see that soil organic carbon is pulling or driving a lot of these properties to the right and separating the subsoil treatments from them, indicating or further suggesting that organic carbon or organic matter is strongly driving the responses we observed at this site. And the regression that was done, the regression that was done was between total microbial abundance and above ground biomass. And it was used as a predictor of, how strong is this relationship? If you can have or recover below ground microbial biomass, would that help achieve greater plant production? We had a positive correlation with an R-squared value of 0.26. And I think it's important to remember that the TDU produced greater biomass, but contained microbial properties or their abundance similar to the SP. And if you relied only on reclamation success on above ground characteristics, you may have incorrectly concluded that the TDU was more recovered than the SP treatment. And that is also why this correlation might have been a little weaker than what it was expected. But it's an important takeaway that looking below ground is also important in gauging the overall reclamation success. So with that, our conclusions were that overall thermal desorption did not impede the recovery of microbial properties. And in some cases, it can increase the potential for biomass production when environmental conditions are met, such as increased rainfall or as a result of the shifts in soil properties. The topsoil was the best chance or the best to use for increasing reclamation success. But if you were in a scenario of topsoil shortage, mixing it with subsoil in the 1-to-1 ratio would also allow soil biological property recovery. Symbiotic relationships between plants and the AMF were not negatively affected by remediation methods. They occurred in all treatments and just were more dependent on soil properties or P scarcity. And also the natural recolonization of the soil microbes were still ongoing. It was obvious that the subsoil treatments were behind the topsoil in terms of biomass, but with additional organic amendments or additional inputs of a source of carbon for these microbes, their recovery could be increased. So we learned some things about microbial recovery there and about how TDU was affecting their recovery and more importantly subsoil. But there were still questions about how else can we improve microbial recovery. One answer might be biostimulants, and they're a pretty new buzzword in the agricultural and remediation field, but essentially that biostimulants are organically derived products that are applied to soil to enhance the natural microbial function or increase plant growth. And some products include humic substances, seaweed extracts, organic wastes like manure, or microbial inoculants. And per definitions from the EPA, they cannot provide a direct fertilization benefit to the plant. And like I said, they're used in agricultural and horticultural settings, but have been increasingly marketed in reclamation sectors. And it's troubling because there has not been the same amount of quantitative data that has been conducted on their use in reclamation. And this lack of research for use in reclamation encouraged us to do a literature review on looking for papers that did in fact study biostimulants use in reclamation, aimed with this main overarching question of can they improve soil biological recovery and help reach reclamation success? Overall, the answer is there is potential for them to help. We looked at as many studies using biostimulants in reclamation settings as possible, but in some cases we had to substitute studies with degraded soil or just agricultural soil. And by degraded, I just mean heavily degraded from intensive agriculture or from chemical properties such as salinity. Humic substances overall had a positive effect on a lot of the biological metrics that we look at, such as soil organic matter content, grain yield, biomass production, and also increased degradation rates of contaminants. The second most common ingredient was seaweed extracts, and there was an overall finding that they contained properties or compounds that are overall beneficial to plant growth and microbial properties such as plant growth hormones and inorganic nutrients, but also sugars and just organic substrates for the microbes to consume. But some unknowns remain. Mostly in the field, do they fulfill the promises that they say they do? And also how effective, if they do fulfill these promises, how effective are the different ingredient types? This is often hard to answer because the private companies that make these products have proprietary ingredients and sometimes questionable marketing material that they give to reclamation professionals or landowners, encouraging that further studies are needed. So with that in mind, we conducted a greenhouse study using 2 biostimulant products and looking at microbial activity and wheat growth. Our objectives were to evaluate how the biostimulant and soil blending affected hard red spring wheat growth. Our second objective was to determine how the AMF symbiosis responded to these biostimulants and investigate how the biostimulants may have influenced microbial activity and their respiration measured as CO2 efflux. We used a randomized complete block design in a greenhouse here at NDSU using 5 soil treatments, 2 biostimulants, and 4 replications. The soils were collected from an active pipeline installation project in Montreal County. And in this picture, you can still see the corridor from where the soil or where the pipeline had gone in. And it was taken from the Wabec upon Sandy Loam map unit. The topsoil was from the 0 to 15 centimeter depth, and the subsoil taken was anything under 15 centimeters. And they were textured and found to be a loam topsoil and sandy loam subsoil. So these soils were transported back to North Dakota State, and upon arrival, they were put through a 10 millimeter sieve and air dried at 25 degrees Celsius in the greenhouse. We used a spectrum of soil blending similar to the thermal desorption study, where we had 100% topsoil, and all the way down to 100% subsoil. And they were blended into different ratios, which were a 1-to-1 topsoil subsoil, a 1-to-3 topsoil subsoil, and then a 1-to-7 topsoil subsoil. And this was to stimulate the effect that might occur in the field where soil mixing during reclamation. The biostimulant treatments included a Levintia SSB liquid inoculant. It had about 15 different microbial species, and it was just the only other ingredient was water. The Profile Proganics mulch was a mulch made up of woody material, seaweed extract, humic polysaccharides, which are just like sugars, and then a no biostimulant control. They were added to their respective soil mixtures. And I do want to go back and say that the mixtures were mixed and added to the pots to equal at the end 2.5 kilograms per pot. To grow the wheat, we let the soils incubate after mixing and inoculation with the biostimulants before planting. And this incubation allowed the microbes in the biostimulants to recolonize the soil and not have to compete with the plants for nutrients in the soil. So after that 2-week incubation period, 10 hard red spring wheat seeds were sowed into the pots and allowed to germinate. There was 15 milligrams of ammonium put into each pot, 15 milligrams per plant per pot. And upon emergence, 5 seeds were thinned from the pot. There were some nutrient deficiencies that were observed, and so we applied 1 milligram of iron chelate to the pots per Dr. Goose's recommendation. And here's a picture of some of what we suspect to be iron deficiency in the wheat. Final sampling occurred at the 5 to 7 leaf stage after root colonization was ensured to be visible. And to sample the biomass and get the wheat out, we took them out of the soil and cleaned the soil off of roots carefully and recovered as much root biomass as possible. This recoverable root biomass was ultimately weighed as a fresh root weight, and the above ground biomass was dried and then weighed as a final dry biomass value. The root colonization analysis followed the same methods outlined in the Tioga study, where we cut them into 1 centimeter segments, stained them, cleared them, and then stained them and mounted them on slides, with 5 passes going over each root segment for a total of 50 observations. To measure the CO2 efflux or the respiration from the soil, we used an environmental gas monitor and soil respiration chamber mounted onto a PVC ring inserted into the soil in the pots. And this allowed for a clean seal to only measure gas coming off or efflux coming off of the soil surface. This was done every 3 days during the incubation period, and then after the incubation period was conducted once a week for a total of 8 readings. Microbial abundance was analyzed the same way as the Tioga study, where they were shipped and analyzed by microbial ID using lipid extraction and gas chromatography and mass spectrometry. We also analyzed soil properties, including soil organic carbon, inorganic carbon, nitrogen as nitrate and ammonium, phosphorus, and then EC. The treatments were analyzed with a 2-factor ANOVA, with the factors being soil blend and biostimulant treatment, and there was a linear regression performed. So similar to our last study, we saw that the topsoil and the 1-to-1 ratio of subsoil to topsoil again produced similar biomass yield, and was found to be the critical blend for achieving a yield similar to the topsoil. Any mixture below a 50-to-50 ratio had similar treatments, but were less than a topsoil. Additionally, we saw that the SSB liquid inoculant produced greater biomass than any of the treatments, and it is thought to be because of increased phosphorus availability, which we will get to in a little bit. And overall, above ground dry biomass was positively related to nitrogen, phosphorus, and soil organic carbon. This is not surprising because soil organic carbon makes up a large part of organic matter, which is important for plant growth and microbial properties. Overall, topsoil contained the largest concentration of soil organic carbon, and that concentration decreased as quantity of topsoil decreased. There were no significant differences between the biostimulant group block, but that is a result of just the study was likely too short a time frame to really observe significant accumulations of soil organic carbon. Going back to phosphorus, we see that the topsoil and the TS100 and TS50 had similar phosphorus availability levels. And additionally, the SSB had the greatest availability of phosphorus as well. This was most likely why we observed the greater biomass growth in the SSB treatment and the TS50 and TS100. Because of that increased phosphorus availability, it helped enable the plant to accumulate biomass and continue to grow. This was also most likely a result in SSB because the biostimulant itself introduced arbuscular mycorrhizal fungi, which are important for the plant and symbiote relationship in acquiring phosphorus and improving phosphorus nutrition to the plant. But it's also included phosphorus solubilizing bacteria in the mixture as well, which ultimately most likely resulted in increased phosphorus availability. Nitrate was the greatest in topsoil as well and decreased as down the gradient of topsoil. It was also the least in the proorganics, which was a little surprising just right off the bat, but looking into it further, it was most likely a result of increased assimilation of nitrate, which is plant and microbial available, their assimilation of nitrate into their microbial biomass. And this was further reinforced looking at microbial abundances and seeing that the proorganics produced or contained the greatest microbial abundance. And so it wasn't a matter of there was not enough nitrifying activity in the soil or conversion of ammonia fertilizer into the plant available form and into biomass, but it was because it was being taken directly from nitrate form into the microbial biomass. We see that topsoil contained the greatest amount of microbial abundance, and that also decreased down the gradient, which is just a function of the organic matter in the soil as well. And lastly, the CO2 efflux showed a similar trend over the trend with microbial abundance, where proorganics contain the greatest amount of CO2 efflux, signaling that the abundance there was actively living and respiring CO2. Meanwhile, we saw in the soil treatments that we saw that any treatment containing topsoil had a significant increase in CO2 efflux compared to the subsoil only treatment. And that was observed to also extend into per soil treatment basis. The proorganics was increasing the CO2 efflux in that soil as well. Overall, from this study, it was evident that the topsoil concentrations were dominating biological responses. This was observed through the wheat biomass growth and also the microbial abundances. And we also can conclude that the 1-to-1 ratio was also successful in recovery relative to the topsoil, and any mixture or soil blend below that 1-to-1 ratio was not as successful as reaching reclamation. Biostimulants influence biological properties and their recovery via microbial processes, but these responses that you observe may be different depending on the ingredient types. We saw in the SSB, it contained microbial inoculants that didn't provide any organic matter or labile nutrients, but it did stimulate increased phosphorus availability in the soil through microbial properties. In the proorganics treatment, it contained organic matter, and it contained some polysaccharides, which were readily available and consumed by microbes, but it didn't really stimulate a response in the soil through microbial processes. It just provided the nutrients for microbes to consume, whereas the SSB influenced the ecosystem of the soil to drive and improve microbial processes and function. And the respiration that we saw, the increased microbial abundance in the proganics treatment, the mulch treatment, did not ultimately improve wheat growth. We saw that wheat performance was improved with the SSB, which was the microbial inoculant. Overall, from my research in a whole, that topsoil and content of soil organic matter and soil organic carbon was the driving factor of remediation and reclamation success. And soils with not enough organic matter or not enough organic carbon were having a hard time recover. And that was evident in the subsoils and anything below a 1-to-1 mixing ratio. Like I said, the 1-to-1 blending ratios were the most beneficial to reclamation success, which is an important takeaway for instances of topsoil scarcity where you can substitute subsoil for topsoil and most likely reach reclamation success. Disturbances which alter soil properties such as thermal desorption are going to influence any land management decisions you have to make. We observed that the increased saturated hydraulic conductivity on the thermally desorbed subsoil altered the amount of water conducting through the soil profile, transporting phosphorus to the root zone and improving plant growth. But that might not be seen every growing season, especially if you have a drought in the growing season, which did occur on the site and resulted in similar yields between the subsoil treatments. Biostimulants overall have merit in improving reclamation success in the short term, but in long-term field studies, these findings aren't as concrete and results are likely going to be different. A potential best case reclamation scenario from these research would be biostimulant application on blended topsoil and subsoil, which would allow for topsoil sources to be conserved and you wouldn't have to face the cost of importing topsoil from a different area to your site. And ultimately continued organic amendments. We did see some significant responses from the composted manure in the Tioga site, but the one-time application in 2013 likely or did not have many residual effects on what we saw in 2019, but continued use of something like organic amendments would likely drive soil biological property recovery. And ultimately just further evaluations of biostimulants, because there's a lot that is unknown, and it's pledged as something that can fix problems in a short time scale and perhaps replace fertilizer, but we don't know if inorganic fertilizer, but we don't know if it's going to require repeated use of biostimulants and how long an application might last in the soil itself. So with that, I want to say thank you. Thank you, Tom. Thank you, my committee, the North Dakota Agricultural Experiment Station, Patty and Steve Jensen, the landowners in Tioga, Kevin Horsager, Joel Bell, Becky, who could have probably given this talk better than I could, listen to me talk about it, my parents, my boss, and anyone else I didn't mention. Thank you for supporting me throughout this whole process. And with that, I'll take questions.
So one thing to state first is that the first study that Zach spoke about has been published in Agronomy Journal. So we do have that one published already. So which is kudos to Zach for working on it, even though with a full-time job and moving and all that other stuff. So thank you, Zach, for being a great student. So do you have any questions for Zach?
I have one. Which is your recommendation for farmers who wants to improve their soil microbial properties?
And repeat the question.
Yeah, so the question was, what would I recommend to farmers that want to just improve their microbial properties in their soil? Not exactly that have disturbed soil, just an agricultural producer.
Yeah, just to.
I would say focus on soil health. And there's been a lot of research with our extension here that involves reduced tillage or minimum tillage. Cover cropping can help reduce soil erosion, protecting the surface from any way that you might lose nutrients or lose soil particles. When you lose soil particles from erosion or runoff, you lose organic matter, which ultimately is needed for microbial properties to survive. And if you can reduce the amount of disturbance through tillage, you can allow a lot of these microbial properties to recolonize and not have to go through that disturbance every year. And a specific microbe would be fungi, which create networks throughout the soil what's called hyphae, and they produce lamelin, which can help protect soil aggregates. And so by reducing the amount of tillage that you might do, you can, in a result, help microbial species protect soil aggregates. So I would say use cover crops, protect the soil surface from erosion or runoff. Thank you.
Other questions? Any questions? And also,
I can check here.
why don't you check the Zoom universe?
Yeah, actually, I have a question. Hello?
Hi, yep, we can hear you.
Oh, yeah, so with the review on biostimulants, was there any concern about like maybe kind of causing like laziness in microbes? I'm just asking because I deal with like macro soil fauna like earthworms, and sometimes if you feed them or make things too easy for them, they won't do anything. They'll just kind of sit there at the top and not actually make any channels. So I was just wondering if you saw anything about that.
Okay, well, no, I didn't see any specific studies that looked at macrofauna. It was mostly microfauna, or just the soil microbes, and no parameters that I looked at were assessing the activity of worms or macrofauna. So I'm sorry that I might not have the best answer for that.
No, I was just wondering if they caused any like laziness in like the microbes or anything like that. Like, did it cause them to be less active at all?
Okay, well, I think the overall effect that I saw was microbial activity increased, and by either improving their function, or perhaps if you introduce inoculants, it's increasing the interspecies and intermicrobial group competition that perhaps would stimulate their activity, because if other microbes are filling in their niche, then laziness would just let them expire. So I never saw a direct trend, but that was my 2 cents on it.
Okay, cool. Thank you.
Thank you. Anyone else online?
This is Matt. I have an online question for Zach. Do you think the selection of a relatively sandier subsoil for your greenhouse control project would have maybe swayed your results on a 1-to-1 mixture ratio being advantageous for the study conclusion?
So the question would be not picking a finer soil, or could you repeat that?
Exactly. Yeah. So if you think the results maybe would be different if it was more of a residual type soil or a finer textured soil done with that study?
Yes, I would. If it was a finer textured soil, I personally would have been concerned about maybe different properties in the subsoil that could have swayed our results. Specifically, that could be like an increase of carbonates, which the sandy loam was a calcareous soil, but with a finer texture, you might have a greater accumulation. There could have been an increase in some like of a clay, which could allow a greater water holding capacity. We didn't see any differences between gravimetric water content between any of our soil treatments. So with a finer texture in those treatments, you might observe a greater water content in the soil pores, which could have increased the abundance of some different microbial groups which live in the water column of soil. And but if it had some accumulation of not positive soil nutrients that may have inhibited plant growth, I think, like sodium or something.
Thank you.
That- You're welcome.
Yep, that answers it.
That was my boss. For anyone wondering.
His raise depended on that answer.
Yeah, it did.
So, thanks for that, Matt. Yeah.
We have a chat question.
Okay, thank you. Okay, well, thank you, Corinza.
Any other questions for Zach?
Hey, Zach, this is Alex Knudsen. Great job, by the way. I have a question for you regarding microbial like inventories. So do you happen to know if we have a really good understanding what we have in North Dakota, you know, in undisturbed or even agronomic situations for like what microbes are currently present? And if that has any implications for like remediation?
Yeah, okay, so is there kind of a baseline of microbial population?
Yeah.
That we could compare to? We actually did, I should have mentioned it. Our relative abundance that we saw in our Tioga site was around 74.3 nanomoles of biomass per gram of soil. And looking at a similar study conducted in grasslands in Western North Dakota, in the undisturbed grassland, they found there was about 80 nanomoles of biomass per gram of soil. So after 4 years, it seemed like there was a trajectory of meeting undisturbed grasslands in the area. The cropland there in Tioga was under no-till crop production, so that may be different if you're in an area under conventional tillage. As far as more eastern parts of the state, I didn't really look at that because all of our soil took place in Western North Dakota. Did that?
Yeah, that answered. Thank you.
Thank you.
You have a
One more chat. Unless it's still a Corinza. No, it's not. Well,
I guess
thank you, Matt.
All right, well, if there's no more questions, Zach, thank you very much.
Thank you, everyone, for coming and
And please drink some of the ice cream on your way out and take some coffee and whatever else. So thank you, Zach.
I'm closing the Zoom. Thank you, everyone who attended.
Stop the recording.